Monsieur Patterson, vous êtes le bienvenu au Liban et au 19e Symposium des gynécologues francophones. Bruce Patterson received his undergraduate training in molecular biology from the University of Michigan. He then received his MD at the University of Feinberg School of Medicine, followed by his residency in pathology. Dr. Patterson has authored over than 100 manuscripts and has over than 100 articles. Dr. Patterson was formerly Associate Professor of Pathology and Infectious Disease and Director of Virology at Stanford University. He currently serves as CEO and founder of incellDx Inc., a growth stage company that has translated his research discoveries into state-of-the-art single-cell diagnostics in cancer, immunology, oncology, and infection disease. Dr. Patterson va nous parler du HPV OncoTect CDX. The stage is yours, Professor.
Thank you, Mr. Chairman. It's certainly a pleasure to be back in Lebanon, which I'm almost thinking is my second home. It's a pleasure to be here. I think I'm uniquely qualified to talk to this group because I'm both a virologist and a pathologist, so I have no bias towards cytology and histology or virology. My goal is to bring it all together to make your job easier. What does incellDx do? What did we do to discover this technology? Well, what we're able to do is simultaneous quantification of proteins, messenger RNA, DNA cell cycle, and morphology at the same time on the same cells. So a few years ago at Urigen, I talked about the slideless PAP and using an instrument where we could see both cytology and do all the molecular biology and see it and encompass it all in one assay. And what we've done here is taken that technology and not put it on a very expensive machine, but put it in a system where we can give you 1-day turnaround time to get all of your comprehensive results on a particular patient, as opposed to waiting for cytology and waiting for immunohistochemistry stains that have to be read subjectively by pathologists, of which I am one. The other thing that's driving innovation in cervical cancer is that new therapeutics are directed at treating pre-cervical cancer changes prior to the development of cervical cancer, and they really require the staging of the molecular changes associated with transformation and carcinogenesis in order to treat at the earliest possible stage. In other words, what's coming along the pipeline with pharma are therapeutic vaccines as opposed to preventive vaccines, antiviral drugs, and one exciting new area is the use of immuno-oncology drugs to enhance the immune system and the immune response against abnormal cells. You know, what I noticed going around the world giving talks, and I especially notice it in my family, is this assumption that we don't have an immune system actually playing a role in what we're looking at here today. And I think that is something that we all have to consider, where, you know, when you have SIN2 or you have SIN3, it's all or none. You're going the distance. That car is driving. But the fact is, there's other factors in play without any interventions that may be influencing that dynamic. And the onus is on us to provide the gynecologist the most information about that dynamic. So number one, you understand what's going on, but most importantly, you can counsel the patient. And for all of us, that's the most important thing. Now, we all have seen slides similar to this. The previous speaker did a beautiful job of showing how the morphologic changes evolve with increasing histology and also increasing his cytology, and also pointed out the fact that indeed When you look at E67 mRNA, it's right here in the abnormal zone. This is where we like to treat. And HPV DNA picks things up way too early in the pathogenesis of cervical cancer to really help in terms of management. What I used to say when I'd go on and give talks, and I still say it, do you want to know if you're infected or do you want to know if you have disease? As a virologist at Stanford, I had to talk about the point to students that we all have viruses. A third of you have cytomegalovirus. Am I going to have you come in every year so I can look at your CMV DNA? No. A third of you have Epstein-Barr virus in your tonsils. That can lead to Burkitt's lymphoma. It can lead to cancer. Am I going to screen every single one of you every single year for Epstein-Barr in your tonsil? No. Why are we doing it for HPV? Okay? What we really want to know is the virus doing something that's going to cause disease? And the advantage that we have in cervical cancer is it's a continuum. There's a very predictable continuum in the genesis of cancer that we can design diagnostic approaches to tell you where you are. The word we use at my company, word we use at Stanford, is staging. I'm not going to show a lot of bioinformatics today, but we're at the point where I can say that patient has a SIN1 before they even have a biopsy. That patient has SIN2, that patient has SIN3. Just by a composite of parameters, by doing multi-parameter detections and putting them into a bioinformatics models and seeing what we can detect. So I submit here that not only do we have to look at what's generating the flow in this direction towards cancer, but we also can't forget about regression and immunity. And we've actually added PD-L1 to our cervical cancer cocktail, if you will, because PD-L1, as you know, in other cancers, is intimately involved in the immune response against cancer. Cervical cancer is no different. I'm not going to present data here today. I presented it at Urgen. We do PD-L1 on cervical cancer, cervical precancer, right? If you're PD-L1 positive, the immune system is ignorant to the malignant cells. If you're PD-L1 not negative, the immune system can still see the abnormal cells. Unfortunately, if you're negative, you don't qualify for drug, but it's a catch-22. PD-L1 is bad because it short-circuits the immune system, and it's bad for prognosis. It's good because it's a target for therapy. Why not in cervical cancer? Well, this is already answered by pharma, who's come out with PD-L1 inhibitors in cervical cancer. My big question to them for the last few months has been, why not cervical precancer as a method of therapy? So again, we need this integration of virology and cytology and morphology to really decide some of these new therapies. The other thing that's always intrigued me is this focus on sensitivity, sensitivity, sensitivity. When I was on the FDA panel back in the late 90s looking at Digene's data submitted for FDA approval, I was intrigued because Digene at the time always talked about sensitivity and negative predictive value. They're the same thing. They never talked about specificity or positive predictive value. Still, if you still go to big conferences, all you're going to hear about is negative predictive value and sensitivity. You're not going to hear a lot about specificity. And you're especially not going to hear a lot about positive predictive value. But the fact is, SIN1 lesions, about 60% regress. Why do they regress? The immune system. 50% of SIN2s regress. Why? Immune system, or maybe they're not being driven by oncogene overexpression. Surprisingly, 32% of SIN3s will regress. What is that telling you? Well, it's telling me that SIN3 is two different diseases. One that's capable of persistence and progressing to cancer, and one that doesn't have the molecular drivers to go on to cancer, and it's going to stay stable. And we know that from looking at women in their 80s in nursing homes who come into the doctor and have SYN3. And the fact is they probably had it for years and it's been stable. This is a very involved diagram, but this is indeed the process to cancer identified by Harold Zurhausen, a colleague of mine for a short period of time. But the fact is, he identified this pathway where E6, E7 and their protein components inactivate tumor suppressor genes. Tumor suppressor genes cause cell cycle dysregulation, which leads to proliferation and progression. Well, why not, like we did with OncoTect 3Dx, detect oncogene overexpression, and then something more downstream like proliferation, where we can actually stage, where are you in this process? We're managing patients sometimes without knowing where we are in this process. Now, Syntec Plus is actually an interesting test because it's P16 and Ki67. P16 depends on E7. Much easier to quantify E7 than P16. And Ki67 is a measure of proliferation, however, not very sensitive, like doing cell cycle. Not to mention, Syntec Plus requires a pathologist to review it. This can be done in 3 hours, and the machine spits this information out. And it all goes back to the genetics driving transformation and malignancy. So there's this intimate relationship between the molecular changes and the morphologic changes. But the fact is, the molecular changes are happening before you see the morphologic changes. So is it enough just to look at cytology? Or is it enough just to look at histology? The fact is, it probably isn't, because the molecular changes are going on and you can't see those with a microscope on cytology or even a colposcope in the clinic. The other important thing is cancer cells don't produce virus. I think that's the biggest surprise to everybody. But did you ever wonder why viral load never made it in cervical cancer screening like it has in other viral diseases? Well, the reason is HPV is a very high viral load disease. That's why the poorly sensitive analytical sensitivity Digene test had never made it for any other target because it had very low analytical sensitivity, but it didn't matter because in HPV infection you made a ton of virus. You made a ton of virus in everything but cancer. So that's why viral load never made it. And if you ever look at Digene's package insert, the sensitivity of Digene for cancer is 93%. 7% false negative for cancer. Gee, if I have a cancer test, I sure as heck want to detect most of the cancer, if not 100%. So again, this is all about test performance and how to use test performance to your advantage in counseling patients. This was a very disturbing study that was presented at Urgen 4 years ago. What they did is they split a sample from 4,000 patients, and they did every single FDA-approved test on those patients, and they looked at the results. Shockingly, the concordance between these 4 tests was 29% for HPV DNA. 29%. What does that mean? Each test relative to the other test has both false positives and false negatives. So depending on where you send your sample, you may get this result, and then you send it the next time for this test, and you get another result. And it has nothing to do with the biology. It has to do with the instrument and the test. To me, that's scary. HPV DNA, this scared everybody. Because the fact is, we are sending it to different laboratories at different times. And then we're trying to compare results of patients who may be going to different laboratories when this is in the background. Okay? So that led a laboratory in the United States to do a study on 1.5 million samples undergoing PAP. They did almost 400,000 HPV DNAs with Roche. They found almost 3,000 HSILs and HSILs with HPV, and they found 8.6% negative Roche with HSIL PAP. Right? Then, of the 63 HSILs that had a negative Roche, half of them were biopsied, 7 were cancer, which is 21% false negative. Then when they looked at SYN3, SYN2, and cancer, the fact is 27% of the HSIL PAPs were either SYN3 or cancer with a negative HPV DNA. That is scary. Okay? We can do better than that. What did we want to do? Well, we wanted to incorporate the actual biology of HPV and the cell in a diagnostic. Cervical cancer is not an infection, but requires it. That's what we need to think about. I'd have people come up to me and say, oh, I have an HPV infection. I'd say, so what? What's it doing? Or they come up with CMV. I'm CMV DNA positive. So what? Do you have chronic fatigue? No. I'm not going to give you $800 worth of ganciclovir because you have CMV. And I'm sure as heck not going to give you acyclovir because you have herpes DNA not doing anything. We need to think about this in a different way and understand the dynamics of gene expression, cell cycle, and morphology collectively. So we did this with a technology called image cytometry. I know it's tough to see, but we could do cytology and the molecular biology all at the same time in cells in suspension. Okay, so you see these beautiful normal squamous cells. This column would be green if there was E67. There's no E67. The nuclear to cytoplasmic ratio is normal, which is what the pathologists look at to see if a cell is abnormal. But we measured it with a machine instead of with an eyeball, and we were able to say this is a perfectly normal specimen. Conversely, this HSIL, which actually is cancer, you see these very bizarre shapes of cells, including this one, which we call tadpole cells, which is a very young frog. But it has this tail, kind of looks like a fish, almost looks like Trichomonas, but it's much larger. E67 overexpression, look at the heterogeneity in the size and shapes of the nuclei. Its nuclear cytoplasmic ratios are high, the area is high, and it's aneuploid by cell cycle. The one thing that Ki67 and Syntec Plus does not do is tell you DNA content or aneuploidy, which are hallmarks of squamous cell carcinoma that are really bad prognosis. OncoTect 3DX tells you all that information in one 4-hour assay. So that we could categorize SYN3s, again, bizarre shapes, E67 overexpression, normals with nuclear cytoplasmic ratio less than 0.25, perfectly normal squamous cells, nothing in terms of staining. Again, tadpole cells, look at the nuclear cytoplasmic ratio of this cell. This is cancer. And this cell, which is also a tadpole cell. Look, an endocervical cell. It's a columnar cell, E67 positive. This was a pre-adenocarcinoma. This one was I knew this was HPV 18. I didn't even do HPV genotyping. Why? Because HPV 18 makes E67 in the cytoplasm. The bottom line is there's so much information at our fingertips, and it's our job to get it to you, and that's what we're doing. So we converted all of that over into a simple dot plot where I don't think anyone's ever seen HPV RNA versus DNA content. But in low-grade lesions, HPV RNA is expressed in the G0G population of cell cycle. And again, you can see that on an image cytometer. On high grades, the oncogene overexpression is in the proliferating and aneuploid cells. It just makes sense. And it only takes 4 hours to do this, to give you the information that you need. Is this L-cell actually have E6, E7 overexpression over here where we have to worry about it and it's going to progress? No. And we can tell you that. So this is what it looks like in the current configuration. This is a normal sample. There's no dots in here. Look at this one, E6, E7 overexpression, 4 hours. You'll get the information. Oops. DNA cell cycle. We can look at proliferation over here. This is perfectly diploid and normal. This one has increased proliferation. The hallmarks of cervical cancer progression are oncogene overexpression and progression. With that, I do agree with Syntec Plus. The thing is, I can do 96 samples at a time in 4 hours on a machine instead of having a pathologist have to look at it a week later. The other thing is also the reproducibility. Everybody talks about test performance, but they never tell you what's behind it or how we went about the clinical validation. This assay, HPV OncoTect, has been done on 500,000 patients worldwide. It's a novel test, but it's not a new test. And we know how to manage patients with it. So it's very linear across a large range of HPV positive cells. There's intersite reproducibility, within run, interoperator reproducibility. All of the things that you want to see in terms of quality control in an assay. And I won't go into this too much, but here's E6, E7 overexpression, no overexpression with proliferation in a high-grade pattern. Here's a low grade where you have E6, E7 overexpression and no proliferation. But the bottom line is, what does this data mean? Well, when we compared incellDx with Roche, Hologic, GenProbe, and others, funny thing happened. We have similar negative predictive values, similar sensitivities. Look at the difference in specificity. This is clinical specificity. You know what this bar is? Clinical false positives. These are all women who go to biopsy and the biopsy is negative. I always ask, how many women do you see with HPV DNA positive who go to biopsy and you can't find anything? You know what? It's pretty common. We talk to a lot of people about that in the States. The other thing is, how can we combine that with morphology and PAP, which we're easily doing here in Lebanon, where you can look at ASCUS plus, so ASCUS, LSIL, ASC-H plus E6, E7. This had the highest sensitivity, the highest specificity, the highest positive predictive value, the highest negative predictive value, and the highest odds ratio of any combination. This is every combination of cytology alone, HPV DNA alone, genotyping, which I'll comment on in a minute, had a sensitivity of 46%. This is the best combination on the market, and it's the combination of cytology and OncoTect 3DX because it gives you all the information you should possibly want, and leaving in the cytology allows you to look at trichomonas and other infections, which is a good reason to keep cytology around. Roche spent $50 million trying to get rid of PAP, and they couldn't. And the reason is you've been looking at it for so long, number one. And number two, there's other aspects of the PAP that are important in terms of patient management. This is the perfect combination on the market right now. Now, in terms of genotyping, everybody asks about genotype. What genotype is it? You know what my rebuttal is? Why do you want to know? Why do you want to know what the number is if it's a high-risk type? Does it matter? What makes it oncogenic? What makes the high-risk types oncogenic? You know what the answer is? Harold Zerhausen would say, it's because of their ability to make E6, E7. So I asked him one time, do I need to know what the number is? And he said in his German accent, yeah, you know what? 16 is probably the only number you need to know because it's the most aggressive. And I said, well, by most aggressive, what do you mean? It tends to transform more commonly than others. And I said, well, Dr. Zurhausen, doesn't that mean it's more likely, higher propensity to make E6, E7? And he said, I guess you're right. Who cares what the number is? If it makes E6, E7 and it proliferates, it's going on to cancer. Right? Right? 16 is the most virulent. But virulence is measured by its ability to transform and go on to cancer. And we've got that covered. Okay? Now, the other thing is age group. Because I was around in the 90s, I'll tell you a story. Digene submitted their data to the FDA in the 1990s, and it had all age groups of women, 21 and over, right? And the FDA turned it down. And why? Because the test performance wasn't very good. As we now know in young women, college-age women who tend to have multiple partners, they have transient infections, they have really strong immune systems, they get infected, the infection goes. That's not a good thing for HPV DNA. The test performance is terrible. So what Digene did is they went back and looked at their data and they said women over 30, the data looks good. So I get all these questions when I'm just at gatherings. Why 30? Have you ever asked yourself why age 30? Like you're walking down the street and you're 29, and the next day you turn 30, right? You're like, oh my God, you're testing crazy. Right? Why age 30? I mean, it's mind-boggling. Like all my friends who turn 30 are like, oh, I have to worry. No, the statistics tell you have to worry. But it doesn't mean you don't have to worry when you're 25 if you live in Chicago, right? It all depends on the demographics. And I know, obviously, the demographics in Lebanon are very different than the demographics in the United States. Here's the other thing that is very demographic-oriented, and that is the issue of screening interval. Everyone's talking about if you do HPV DNA and it's negative, you can wait 3 to 5 years. I think that's the most dangerous thing you could possibly do, depending on where you live. Okay? And I'll tell you why. Number one, the data I just showed you on the imperfectness of HPV DNA and its relatively high rate of false negatives that nobody ever considers. Nobody ever thinks about the fact that HPV DNA might be a false positive, right? Or a false negative. Well, the fact is, it can be both. Okay? The other thing is these studies with long screening intervals are done in like Kaiser Permanente in the United States, which has thousands and thousands of patients, right? Who pay really big money for insurance. So that population is biased. The prevalence is low. And so, yeah, is long screening intervals good in that population? Probably. But you go to my hometown of Chicago, and you do a 3-year screening interval, where I used to see women with cervical cancer at 19, 20, 22 years of age. No, that doesn't work. So the test performance, the surprisingly high level of false negatives in HPV DNA, and the bias of some of these studies on screening intervals scares me as a virologist. Okay? So the other thing I've always noticed in my 30 years of pathology are these algorithms for monitoring patients for cervical cancer look like a spider web. I put them up on the wall and it just looks like a spider web, right? And if I was a gynecologist and I had to sift through all these sheets to see, okay, which spider web am I looking at for this diagnosis? That would be difficult. And I think there's ways to simplify it when you incorporate both the cytology and molecular analysis. And that's what we did here, is we came up with an algorithm with OncoTect 3DX, which is the only test in the market that has 3 biomarkers. It has E6, E7, and cell proliferation, which are the most important. It'll also tell you aneuploidy. If you see aneuploidy on that test report, it's going to say, go right to colposcopy. Do not pass go. Do not go on holiday. Do not go on vacation. Go right and have colposcopy, okay? But we see with different cytologies, you can do, is it E6, E7? Yes, colposcopy. Abnormal cytology, yes, but no proliferation, retest in 6 to 12 months. Abnormal cytology, no E6, E7, but proliferation, probably an infection. Retest in 6 to 12 months. Cytology, negative, routine cervical cancer screening. Ask HCIL, AIS, positive, you go right to colposcopy, right? And DNA positive, and if it's negative for E6, E7 proliferation, retest in 12 months. Relatively simple chart you can have on the wall. You don't have to have multiple spiderweb diagrams, and you can get to the answer quite quickly. In summary, there was a great quote by a colleague of mine, Phil Castle, which I think was visionary because it was back in 2010. But it was in a time when everybody talked about sensitivity and negative predictive value and forgot about everything else. That finally there must be a greater appreciation that both clinical sensitivity and specificity, or their complements, false negative and false positive results, are important to patient safety. In other words, with HPV DNA with a sensitivity of 96, 97% and a specificity of 20 to 30%, and you only do that alone, that's dangerous. Quick story from when I was doing HIV, and we first came up with the ELISA in the late 80s and early 90s. The test performance of the HIV ELISA was 99% negative predictive value, 20% positive predictive value in specificity. You know what we did in the clinical labs then? We wouldn't report out a result unless we did a Western blot, right? We wouldn't even report those results out. The HIV ELISA is the same test performance as HPV DNA, yet we report that result out and we act on it without doing a confirmatory test that has the specificity that we need to ensure patient safety. Anyway, I want everyone to consider that the combination of morphology and OncoTect 3DX is the perfect combination in this setting that gives you the proper balance of sensitivity and specificity while detecting potentially other infectious diseases that you might show up on the Pap sample. So I'll leave you with that. I know I've gone over a lot. It's probably a lot different than what you've heard before, but trust me, things are changing. And the next thing that's going to change is we're going to have therapies for pre-cervical cancer. And we're going to start next year, I guarantee you, next year, we're going to talk about immuno-oncology in terms of cervical cancer. So be ready for it. And be ready to start thinking about, with your patients, the fact that they actually have an immune system, okay? If I can leave here today and everybody's thinking that they actually have an immune system that's factoring into what we do, I've done my job. Thank you for your time. I really appreciate it.
Thank you, Professor, for your valuable intervention. I leave the floor for the questions.
In your experience,
Yes.
no cancer can be produced if E6 and 7 are negative according to your test?
There, as you probably know from Zurhausen's work and others, 0.5% of cervical cancer is caused by P53 mutations. So yes, 0.5 to 1% is caused in the absence of E6, E7 expression.
All right, and according to your test, what will be the false positive and false negative?
Well, again, the false positive and false negative rate for histology is, you'll see a diagram at the booth. The false negative rate is about 2% to 3%, which is the same as HPV DNA. However, the false positive rate, our specificity is 85% versus 20% for HPV DNA. That's where we really shine in adding this huge increase in specificity to the equation. And what that does is it pushes our positive predictive value over 50%. There's no other test on the market that has a positive predictive value of greater than 50%. HPV DNA's positive predictive value, there was a test by Hologic called Cervista a few years ago. Their positive predictive value was 8%. 92% of women who had a positive result did not have SIN2+. Excuse me.
Thank you very much for this presentation. As I understood that age is no more important below 30 or above 30. If a patient is a 25-year-old and she has, for example, ASCUS or LSIL, would you directly go for E6, E7 proliferation?
Come to the booth because I went over that one table very quickly, the one by age. We have stratification from 20 and over, 25 and over, 30 and over, the test performance. And OncoTect 3DX has higher sensitivity than HPV DNA for women under 25. So yes, age is extremely important because of the transient infections, and we need to find a way to be able to monitor those patients appropriately.